Good morning. Happy Friday, Brodies. I'm Bronwyn McGinnis. It's an honor to host this installment of the Broads IDMP seminar series as it continues to highlight important perspectives on the COVID-19 pandemic. This morning, we have a truly exciting series of short talks on a very hot area of research into the single-cell genomic analysis in acute, severely infected COVID patients, highlighting some of the really cool and very Brody science and collaboration across labs, institutes, and hospitals in our community. Dr. Alexandra-Chloe Villani is an assistant professor at HMS, the director of the Single Cell Genomics Research Program at NGH, and an associate member at the Broad. And she will give an overview of efforts of her team and collaborators on COVID single cell work. Then Toni Delorey, a research scientist at the Broad in the Regev Lab, and Carly Ziegler, an MD PhD student in the Shalek Lab, will follow up and take us through their projects and findings. And then Alex and Arit will join us for questions. It's a stacked agenda. It's surely going to be a great talk. So I'm just going to hand it over to Chloe now to get us started.
Thank you so much for the introduction. And first and foremost, Toni, Carly, myself, Alex, and Arit want to thank you for inviting us to share some of the highlights of the work we're doing in collaboration with several hospitals across Boston, including NGH, Brigham and Women, and Beth Israel. This is also work being done with several colleagues across the Broad Institute and, of course, the Climate Cell Observatory. And so the scope of our work takes place because we know so little about the immune and cellular response associated with COVID infections, the range of symptoms, and lethality. But we know that some patients develop severe disease in the lung, such as acute respiratory distress syndromes with some uncontrolled inflammation and cytokine storms, but yet we don't know why. We also know that beyond the lung, many other organs can be affected, but again, we have very little understanding of why this is the case. And finally, some people will go on to develop mild disease and others very severe across different organs. And once again, we know little about this. So this is why we decided to try to leverage the power of single-cell genomics analysis to start answering these questions. Before we start going into greater depth about our analysis, we wanted to make sure that everyone understood why we go at the single-cell level to do our analysis. Of course, the COVID-19 can affect several organs of the system, but as we know, the system comprises several organs which are made out of tissues, and within tissues are captured many different single cells. Some of these cells are infected by COVID-19, some are bystanders. And so we want to understand which one of these cells are affected by the dysfunctional immune system. And so to do so, we are trying to use a data-driven approach to be able to map all of the different type of cells that one can find within a tissue. So how do we do that through this single-cell approach? Of course, here we are oversimplifying the concept, but the idea is that through our DNA are encoded a set of genes from which are translated mRNA, and there are unique transcriptional features that are expressed in different cells. And so we are using these unique transcriptional features to define the range of cell type and cell states found in our body, and thus we can use the same approach to identify those that are infected by COVID-19. With this approach, we are capable of identifying those that are infected. We are capable of identifying the cell composition of different tissues. We are capable of figuring out which cells are likely to interact with each other, and we can do so through a range of single-cell toolbox that has been developed across our labs. That includes some single-cell and single nuclei sequencing, where we can capture single cells in drops to derive such information. We can also do such analysis in situ, where we can map within the tissue the single-cell transcriptome to start understanding which cells are talking to each other. We can use different type of computational approach to group the cell. Here, every single dot represents a different cell, and we can regroup them according to these transcriptional features that they share to start understanding the communities of cells that share similarities. We can then leverage the data from our single cell analysis to try to derive unique pathways that are enriched in these infected tissue in order to identify new biomarkers and therapeutics. Across our several efforts, we are spearheading projects that are focused on the blood, including understanding the drivers of the acute forms of the disease. I will talk about this morning. We also have an effort around convalescence patients, so we can identify neutralizing antibodies. There are efforts around tracheal aspirates, nasal swabs, and bronchoalveolar lavage analysis. And finally, we also have a large autopsy effort where we have the opportunity to do a deep dive in understanding which cells in the tissues are affected and are driving the host immune response, which Toni and Carly will talk further about. Some of the questions we hope to answer includes what are the major players driving the host immune response, which cells are affected in the tissue, what is the direct impact in infected cells and bystander cells, how does disease phenotypes vary across similar cells in different tissues, and finally, can we find novel pathways targeted by the virus that will lead to the identification of such biomarkers and therapeutics. So I'm gonna give you some highlights of one of these efforts. It is an acute cohort of patients that were recruited in the emergency department at MGH under the leadership of Michael Philbin. These are the different investigators at MGH that are involved in this effort, as well as Nir Hakohen and Paul Blaney at the Broad Institute. During the scope of 6 weeks, we've recruited any patients presenting at MGH emergency department with acute respiratory distress syndrome. And here you have the breakdown of the patients in a range of severity, which we have further classified according to the World Health Organizations at the bottom here. And you can see that we have some severe patients, some of them that have unfortunately passed away, some that had to be intubated, and some that recovered or did not recover oxygenation. A portion of our patient had severe complications, including cardiac complications, as well as severe lymphopenia. Within these patients here, those that were hospitalized, we managed to collect samples at the 3rd day of their hospitalization, as well as their 7th day of hospitalization. And in some cases, we also collected the blood samples in the context of what we call an event trigger, which could be, for example, intubation or extubation. In the scope of 6 weeks, we've collected over 788 peripheral blood mononuclear cells, or PBMCs, as well as plasma and bulk neutrophils. And here on the right, I'm highlighting the work of 20 superheroes who work 7 days a week, 5 a.m. to 2 a.m. in shifts to be able to recruit these patients and process the blood fresh. This was no small task, as you can imagine. And so what are the planned measurements? So for all of the immune cells that we have biobank, we're performing single-cell RNA sequencing with paired measurement of 197 surface protein, as well as paired T-cell receptor and B-cell receptor sequencings, and are planning from some functional follow-ups. For neutrophils, which are more difficult to capture by single cells, we've isolated fresh a minimum of 100,000 neutrophils per samples and are performing bulk sequencings for 10,000 cells for each of these 788 PBMC samples. And finally, we've also biobank plasma, on which we are performing a range of proteomics analysis, including the analysis of up to 5,000 targets. We are also doing virus neutralization assays, testing antibody reactivity and properties with different collaborators, assessing the role of pyroptosis, and again, doing a range of other functional assays. And here I'm highlighting the amazing analysis teams that bridge between, in this case, the Broad Institute and MGH. And so some of the questions we seek to answer specifically for the blood analysis is understanding which immune cell states are associated with each of these disease subgroup, which immune cell states are associated with the different phases of the disease, as we have different time points from day 0, which actually is not day 0 of the initial infections. By the time patients are admitted, they're usually around day 8 of their infection. We also want to understand the activation cell states of the expanded T and B cell clones as compared to the non-expanded clones. We want to understand if there's new clones at each phase of the disease over time. We also want to understand if there are innate immune response that are reprogrammed during the early phase of the disease that can then lead to altered response in the later phase. And finally, we want to understand which particular immune cells are contributing to the cytokine storms in these severe patients. And so over the last few months, we actually had to overcome several challenges. Some of you may think, oh, analyzing BIDMCs is pretty straightforward. In this case, it was not so straightforward because, as you saw in some of the previous slides, patients were lymphopenic, and we got very little blood from these patients as they were very sick. On average, we got more around 3 to 4 ml. And so we had to come up and develop new miniaturization cell isolation assays, and we will be releasing such protocol. The second type of problem was lower cell survival when we taught these vials. And this was more related to the fact that these patients were suffering from acute inflammation leading to more cell death. And so here again, we developed new protocols to improve cell yield and deplete the dead cells. And finally, we have a large number of samples to profile, each with very few cells. And so we needed to find a way to scale up the processing of the samples for single cell analysis, which we did. And I want to make a shout to Miguel Reyes, who's a very talented graduate student, co-advised by Paul Blenny and Nir Cohen, who took the protocols that is normally done in Eppendorf tubes and modified it to allow processing to now be done in 96 well plate. This is usually transformative for the entire effort, especially since every time we perform an experiment on one patient samples, we need to thaw 4 vials. And so without going in great depth, because this will be released publicly with our data, Miguel has developed a protocol that 2 people can do in parallel, allowing the processing of 8 samples, and you can do this processing in parallel, so you can actually do 16 samples in parallel, depleting dead cells and red blood cells, enriching for immune cells on the left, and for B and T cells on the right, using basically magnetic beads principle. So kudos to Miguel. So we have some initial preliminary data. I have to say that the last few months I've been spent at developing the protocol I just alluded to. So here you have a representation of 10 different patient samples, and we can identify the different cells that we see here. We see T cells, right, as if it's green, the more green it is, the more particular gene is expressed. And here I should say that every dot is a single cells, and they're regrouped according to the communities of cells that share transcriptional features that we talked about. These are NK cells. You can say that because they don't express the CD3 markers. You can identify the CD8 cells, the CD4 T cells, the B cells and the plasma cells, as well as our myeloid cells. And now we can look at if there's, in this preliminary data, any particular cell subset that are enriched in our cases through these density plots. So the more red it is, the more particular cell state is enriched in case or control. And we can see that just by eye, we have some enrichment of some myeloid cell states in these severe cases and some of these CD8 T cell states. And we can start looking at what exactly these cells express. And that involves, for example, and unsurprisingly, this is known, that some of these CD8 T cells start expressing some of these exhaustion markers, of course, because of the continuous antigen exposure. They also express, interestingly, some of these trafficking and retention markers, such as CX3CR1 and IDGAL, suggesting that some of these T cells may be trafficking to the tissues and amplifying the host immune response. In terms of the myeloid cells, here at the far left, we can see that some of these myeloid cells express different markers of the inflammasome pathway, as well as interferon and interleukin-6 related genes being expressed here at the bottom. And finally, they also express homing factors that could help, the analysis of which could help us understand which cells ultimately traffic to the tissues and can lead to patient's demise. Miguel, again, talking about some of the work from Miguel Reyes in our group, had recently published in February a paper in Nature Medicines studying the signatures of bacterial sepsis at the single cell level, and had identified within his monocyte population, particular population here highlighted in red, enriched in sepsis cases that I further highlight here with this red arrow called MS1. And interestingly enough, that particular population was associated with severity of disease and organ failure. when we look at our own myeloid cell population and recluster that population, we interestingly can also see the same score, which is called MS1 for this MS1 state here in sepsis. And interestingly enough, this particular score does correlate with severity of the infection in this patient population. Of course, this needs to be taken with a grain of salt, as this is just our initial pilot experiment. And so moving forward, we are now ready with our protocols. We are starting to scale up our data generation in the coming weeks. We are seeking to perform single-cell multi-omics analysis on this entire cohort I just introduced you to. That includes these 306 COVID patients across the different time points and clinical subgroups I've introduced you to. We will include some COVID negative patients and some healthy controls that will match the demographics of these patients for a total of close to 800 samples. Some of the more advanced analysis we'll be performing, including trying to integrate our paired TCR and BCR data with our single-cell RNA sequencing data to map these expanded clones to specific lymphocytic states. We will be integrating the surface protein data we've been generated with our single-cell RNA sequencing data to refine our cell state definition and classification. We will also start modeling the different cell states over time, so we can try to understand if there's particular cell states that are associated with outcome. And of course, we'll be comparing the different cell states with different metadata such as cardiac event and D-dimers. Ultimately, our goal is to unravel the cellular response to virus to find new therapeutic and biomarker targets. So I've mostly talked so far about defining these cell states using single-cell RNA sequencing, bulk neutrophil analysis with site-seq, and this TCR and BCR analysis. But ultimately, what we're going to do is integrate additional modalities of measurements that I've introduced, including secreted factors that may be associated with dysfunctional host response that we are measuring within the same patient samples, as well as deciphering the antibody properties and reactivities, measuring the virus neutralization. The last thing that I'm going to introduce is that for some of these patient samples, or some of these patients unfortunately passed away over the last few months. And so we teamed up with some pathologists at MGH to start collecting these samples, for which we now have both paired blood and tissue. And this is what brought us to also team up with Alex, Orit, Carly, and Toni, as well as other members of the Boston White community to perform autopsy analysis. And so we want to understand why certain patients enter a second phase of disease that can lead to death. Is it a change in the innate or adaptive immune response over time, right? So it is a strength to be able to compare paired blood and specific tissue to understand if we're looking at the same immune cell states driving the disease. And so for the paired analysis, we are specifically focusing on lung, liver, kidney, heart, and in one case, brain, but Toni and Curtis will tell you more about this. And so beyond trying to further understand this host immune response associated with patient demise, we will be finally able to see if there's anything that is mimicked in the blood that could also serve as a biomarker, and if the immune cell states are altered by the initial virus challenge that can leading to aberrant response at the new tissue sites, and whether we are dealing with different or new TMP cell clones associated with tissue damage. So with that, I will let Toni now tell you more about this important autopsy research effort that actually bridged between MGH, Brigham and Women's Hospital, Beth Israel, and the Broad Institute, and other partners across Boston.
Thank you, Chloe. So the work Carly and I will talk about today is the effort to profile organs collected at autopsy from COVID-19 patients. So studies like this are really unique in that we're able to collect multiple tissues across donors and interrogate COVID-19's effect on each organ. So this is what we'll focus on today. And with the tissues from these COVID-19 donors, we have three main objectives. The first is that we'd like to build a cell atlas of SARS-CoV-2 infected tissue that will describe cell composition, cell state, and cell interactions. Second, we'd like to compare diseased versus healthy tissue and ask how cell compositions change during infection. And third, we hope to be able to assess what cell types tend to be infected and what gene expression programs are turned on in these SARS-CoV-2 infected cells. So in order to answer these questions, we can use the cutting edge technologies that Chloe mentioned, like single cell and spatial RNA sequencing, which will provide a high resolution view of these infected tissues. So we were able to quickly leverage our network of Boston area hospitals and adapt our existing infrastructure and protocols that we normally use to profile clinical samples to process these SARS-CoV-2 infected tissues. And because the disease has been shown to affect multiple organs, we've actually collected many tissue types that are listed here, including multiple portions of the heart and the lung, the lymph nodes, trachea, and spleen. And we now have tissues from 17 donors and over 420 tissue specimens. As Chloe mentioned previously, this is really a multi-institutional effort, and this work really wouldn't be possible without teams at MGH, BWH, and BI. And we're very thankful to all of our wonderful collaborators here at the Broad and at these Boston area hospitals. We, like Chloe, faced challenges when we began this work, and there were three main ones that I'll talk about today. The first was sample coordination and processing. The second was adjusting tissues and processing protocols, and the third was adjusting our computational protocols. So our first challenge was sample collection and coordination. The team really needed to be alerted to the possibility of tissue allocation and really promptly communicate with the physicians and all of our team members across institutions. So we actually did this via Slack. We found that we could communicate in real time about when packages were arriving, and if Carly and I had any questions, we could communicate with the team actually from within the BL3. We needed to make special kits to send to the pathology lab so that they could send us samples safely and quickly and keep the tissue in good condition. And we also, importantly, needed a space to safely process these tissues that presumably some of them carry high titers of virus. So after multiple discussions with Broad's EH&S team, it was decided that these samples needed to be processed in the BL3 facility at the Broad, and that's normally reserved for tuberculosis research. So all of this was happening when the Institute was actually closed for everything except COVID-related research. But thankfully, we were still able to connect with Deborah Hung's lab and use her group's BL3 space. But this required us to undergo rapid training to work in this high safety level facility. So James Gomez, Zohar Bloom-Ackerman, Tori Tran, and Alex Sturm of the Hung Lab were really instrumental in helping myself and Carly Ziegler, who will talk next, get up to speed and begin processing these samples. And what Carly and I saw immediately was that there was actually gross heterogeneity in the tissues that we were receiving among donors, especially in the lungs. So here at the bottom, I'm showing 6 pieces of lung tissue from 6 different donors that look very different from each other. And this table really highlights the variety of tissues that we sampled from each donor. And it also shows how donor age varies really widely. So we have patients in their 30s and patients in their 90s, for example. It shows the organ failure experienced by the donors, which also varied from multi-organ to lung to kidney failure. It also highlights IMV, which is intermittent mandatory ventilation days, meaning the days that the donors were on a ventilator. And it also shows the postmortem interval, and that means the time that has passed from the time of death to the time of autopsy, and that ranged from 1.5 hours to 24 hours. So these long PMIs were really daunting, given that we're actually trying to measure single cells and RNA from these single cells, and RNA can actually degrade pretty quickly in dying cells. We recently received a set of samples with much shorter PMIs from rapid autopsies, and we've begun to profile those as well. We began by profiling single cells from the lung as SARS-CoV-2 attacks this tissue in most infected patients. And to do this, we both dissociated tissues into single cells as soon as the tissue arrived and then profiled them immediately. But we also profiled biopsy-sized portions of the tissue that we froze viably. We now have 17 donors and 420 tissue specimens, as I said before. So here we're seeing data from 7 lung samples across 3 donors, including from fresh cells and cryopreserved cells. And on these UMaps, as Chloe mentioned, each dot is a cell, and cells with similar gene expression profiles cluster together. And then we can look at those clusters and identify the types of cells there. We were really delighted to see that we were able to identify biologically relevant cell types that are normally found in the lung, including AT1 and AT2 cells. We also saw immune cells like macrophages and T cells. But when we looked at the UMaps of the individual samples, which I'm showing towards the right of the screen, and then compared them to the UMap of the combined data on the left, it was evident that not all cell types were found in each of the individual samples. So we adjusted our tissue processing protocol and next tried single nucleus RNA sequencing on flash frozen biopsy-sized pieces of tissue. This method has been shown by our lab and others to capture sensitive cell types that can actually be lost during the lengthy processing required to obtain single cell dissociations. So here I'm showing you data from 8 single nucleus samples that we profiled across 7 donors. And again, we're able to see biologically relevant cell types of the lung. And this time when we looked at the UMaps of the individual samples, we could see that almost all cell types were present in each sample. So the computational experts in our group were able to capitalize on workflows they already had in place for other projects involving clinical samples. And this helped us quickly evaluate the success of our sample processing. They were able to adapt these workflows to better accommodate a really challenging data set. So just very briefly, all samples are demultiplexed and they're then aligned to our new human and SARS-CoV-2 reference, which Carly will discuss a little bit later, to generate a counts matrix. We then apply CellBender to remove ambient RNA, which I'll touch upon briefly next. The data are then further processed to evaluate quality, remove low quality cells, and cluster using Cumulus, and that's a cloud-based pipeline developed by Bo Lee and his team. And in parallel, we also assess what the data looks like without applying CellBender to ensure rare cell types have not been inadvertently discarded. And then finally, cell subsets are annotated using, we use automated methods, known gene signatures, and manual annotation of marker genes. So as I mentioned before, when cells die or are degraded, RNA can leak out of the cell, and this can cause background and gene expression that we measure in our single cells. So to mitigate this, when tissue is normally processed in our lab, it occurs as quickly as possible after resection. But as I mentioned, and as you saw in the table, these samples were collected up to 24 hours after death and autopsy, and then sent to us for further processing. So because of this, we expected high levels of ambient RNA. We also saw the gross heterogeneity in tissues that we were receiving across donors. We have these long PMIs, and now these expected elevated ambient RNA levels meant that this data really presented us and the computational experts in our group with some big challenges. So luckily, Maritosh Babadi of the Broad actually had developed an open source software package to take on common quality control issues in single cell and single nucleus data, including ambient RNA. And he actually, with the help of others in our group, modified this package for us. And with this tool, we were able to remove low quality cells and ambient RNA from our data set. And that really led to more defined cell type clusters that I hope you can see on the right. The next tissue we've begun to profile is the heart. Studies have shown that the virus can be detected in the heart tissue, and heart failure can be a complication of COVID-19. So just like with lung samples, we found that CellBender significantly increased the quality of our clustering. And this is data from a single heart sample taken from the left ventricle. And here we were again able to identify biologically relevant cell types. And again, in this, these UMAPs here are actually from the same sample, but a red or a yellow color means that a cluster is enriched for the cell types listed above the UMAPs. And now we're in the process of profiling all samples across donors, including heart and lung samples. And we really want to understand how COVID-19 affects these organs. Sorry about that. So Carly and I are very excited that we've now processed to completion one portion of the heart and lung for all donors. We're in the middle of beginning to profile the kidney and liver, and we're going to try to get data from more challenging tissues like the spleen and the lymph nodes. And hopefully this will allow us to get more information about immune cells, which will be really nice to integrate with the data from the acute blood cohort that Chloe discussed. We're also starting to build a collection of non-COVID infected tissues, and we're going to use rich data sets that are already out there, like the Human Cell Atlas, to use as controls for this data set. And now Carly will discuss other dimensions of analysis, including viral co-detection and spatial approaches that we're using with these tissues.
Great. Thanks, Toni. All right, so as Toni detailed, in the past few months, we've really developed and put in place these complex protocols from sample processing to the computational pipelines that have allowed us to create these really growing data sets. And so an important question that we're now equipped to ask is which tissues here represent direct targets of viral infection and therefore harbor virally infected cells and are potentially sources of viral replication and transmission, and which other ones experience dysfunction during COVID-19 disease through potentially other mechanisms such as inflammatory damage. So to address this question, we're taking a two-pronged approach. So first, utilizing the host viral co-detection from our single cell and single nucleus RNA sequencing experiments, we're able to unite the presence of cell-associated viral RNA to the host cell identity. And second, using highly multiplex spatial approaches on tissues from these same donors, we can identify viral products within cells in their native environment. And so with this, we can really start to understand the cellular interactions that occur between host and virus and bystander and infected cell in situ. And so really, thanks to this huge global effort to better understand SARS-CoV-2, we were able to leverage an existing detailed characterization of the sequence diversity and the structure of the viral genome to build our reference for alignment. And so here we're taking also advantage of the fact that SARS-CoV-2 is a polyadenylated virus. And so standard protocols for single cell, single nucleus RNA sequencing are therefore equipped to capture viral RNA as if it were just another transcript within the cell. And so we built this pipeline for joint alignment to both human and viral genomes. And we first confirmed that this approach is working as expected by testing against a set of pre-COVID samples from various tissues, which here serve as negative controls. We also tested this from data from human bronchial epithelial cells following in vitro infection with SARS-CoV-2 isolates, here serving as a true positive. And so after some tinkering with our alignment references and the parameters here, we recovered these results which really clearly distinguish virally aligning reads from these single cell genomic data sets. And so what was incredibly interesting and exciting was that when we looked at the COVID-19 autopsy tissues we collected, a small subset of which are shown here in red, we found specific cells containing high abundances of cell-associated viral RNA. And so this is independent of other metrics such as cell quality or sequencing depth. And so what this allows us to do is ask specific questions about which aspects of the clinical history, the disease progression, the tissue of origin really associate with viral replication. And so I'm going to highlight some very preliminary, but some interesting points here is one, not every tissue, even not every lung region within a donor who has unfortunately died of COVID-19 contains high abundances of cell-associated RNA, viral RNA. And so we're really just beginning to map out the heterogeneity between individuals, even though all of them in this study have severe COVID. Two, in some of these samples where we do find higher viral reads, we can ask if there are any trends or commonalities that may correlate to this high abundance of virus, such as for these being very early in the disease course, a few days since the onset of symptoms. And third, what we can do is ask how widespread viral infection is throughout the body. How many different anatomical sites can we detect cell-associated virus? And so an additional important note for this analysis is that we need to be incredibly careful in how we interpret the presence of viral reads. And so single cell, single nucleus RNA sequencing are inherently plagued by this problem of contaminating ambient RNA. And Toni mentioned some of the really amazing computational tools that are being created and really leveraged to address this issue. And so far, what we can do is build classifiers and heuristics to call cells as virally infected versus bystander in these tissues by analyzing, among other things, the viral genomic properties, such as the location of the reads across the viral genome, the diversity of those regions within single cells, strand alignment, etc. And so finally, beyond looking at the tissue location or the overall abundance of the virus, we can connect the identity of the single cell, so the specific cell type, the specific cell state, to their infection status. And so on the left here, I'm showing you a UMAP representation of over 30,000 single cells across a variety of donor lungs. These are named by their presumptive identity at a course level. This data enables us to map the expression of cellular factors for viral entry, such as ACE2 and associated host proteases. As expected, these are generally found within epithelial populations, although not strictly. And we can further overlay onto this map the viral RNA status of each single cell. So viral positive cells are shown in black on the right. And so this data really allows us to ask some key questions. First, what's the identity of the cells that contain viral RNA and how these may change across donor tissue? Two, what are the associated gene expression programs among directly infected cells or unique antiviral or inflammatory properties of the myeloid cells that we actually see potentially actively engulfing or clearing infected cells? And third, we really want to assay the capacity of virally infected cells and bystander cells to interact or communicate directly with one another. And so this may illustrate both the cell intrinsic response to virus as well as the bystander immune reaction. So this dimension of cellular interactions and cell context leads us to the next part of the talk and kind of the next section of data. And so I mentioned that the single cell or single nucleus transcriptomic data is really only a small portion of this work. And so an absolutely essential complement to the single cell genomic side are these in situ maps of COVID-19 impacted tissues. And so with these, we can link the presumptive cellular harbors of viral infection to their native spatial context. This work, we hope, will yield a deeper understanding of the cellular interactions, the landscapes of severe COVID. And so we're approaching these FFPE-banked tissues with multiple complementary approaches for spatial analysis. And so some of the really incredible team that's spearheading these efforts are listed here below. And so one such approach is the nanostring genomics assay. This allows us to zoom in on specific regions of interest within fixed tissue and measure cellular components, including both RNA and proteins within that region with very high multiplexing capacity. And this enables cellular markers within the thousands. Through segmentation analysis of specific regions and subregions of interest here, we can analyze infected tissues with high spatial specificity, which I'll detail more in the next slides. And so here, we're looking at trachea tissue from a COVID-19 study participant. In this particular experiment, 1,800 RNA targets were assayed within 19 regions of interest that were pre-identified within this tissue. These regions were chosen based on binding of an RNAscope probe that targets SARS-CoV-2 viral RNA. And on the left here, nuclei are identified in blue, and in bright pink are SARS-CoV-2 RNA high cells. Mostly these are found residing within the tracheal epithelia. On the top right, you can see a zoom in onto two of those areas. And so using this approach, the team can now mask and divide each ROI into those subregions that are either SARS-CoV-2 RNA high, shown at the bottom in green and yellow, and directly compare those to abutting areas that are largely negative for viral RNA, here shown in white. And so from the same donor, here I'm showing you lung data following the staining, the segmentation, and analysis is done before. Here we include ROIs from multiple alveolar, airway, bronchial duct, connective tissue, blood vessel regions. And so just kind of at a high level, the few early takeaways from these analyses is, first, we see very high viral load in some portions of the lung. And this really just highlights how extensively SARS-CoV-2 can attack and replicate within this tissue. And this is in agreement with our single-cell genomic data from this same donor. And second, we can see areas of high viral abundance directly adjacent to sections of the lung with very low viral infection. And so the diversity of both cell targeting and the regional heterogeneity we're only starting to make sense of. And really, together, our aim is that these spatial analyses and these genomic approaches will mutually inform each other. And so this will hopefully help us understand which host cells are more susceptible or more targeted by viral infection than others, and how this relates to the bystander immune response. And so up until now, we've walked you through all the layers and work in progress that enable us to build this growing data set. And so I'll leave you with a summary of what this effort is trying to answer. So first, we want to build a cell atlas of SARS-CoV-2 infected tissue. And this work is really focused on these exceptionally severe cases of COVID-19, where the participant sadly dies from their disease. And so I also want to mention here that we're incredibly grateful to these study participants and their families that really have enabled this work. And so at the most basic level, we'll be describing the cell composition, cell state, interactions, in COVID-19 across tissues. This will give us a census or a landscape of the disease. Next, given these incredible reference data sets that have been generated by efforts such as the Human Cell Atlas and other research projects, we can compare COVID-19 disease tissue versus healthy samples, and we can put this in context of, say, other types of viral pneumonia and tissue pathologies. And so here we can ask, what are the specific changes that are unique to COVID-19 infection or common host responses across different types of disease? And finally, and I showed you some preliminary data to this point, is we're hoping to ask what cell types are infected, which are responsible for replication and transmission of SARS-CoV-2, and what are the gene expression programs that are either induced or repressed in virally infected cells. And so really, most importantly, our plan is to share our data rapidly and openly with the entire community. And so we have many different sources for making this data available and queryable after some pre-processing. And so this should be accessible for both those with extensive computational backgrounds, as well as newcomers to genomic analysis. And this data should be available soon. And so finally, as this epidemic continues, we really hope that this work will fit into a united perspective of the biology of COVID-19. In this particular study, we're asking questions on the impact of severe disease, and given the large blood cohorts that Chloe highlighted at the beginning of the talk, we may be able to nominate potential risk factors that discriminate severe versus mild infection. We're also involved in mapping the tissue response in pediatric patients. And so the motivation here being that, incredibly thankfully, younger cohorts tend to get less severe COVID infection. And if we could understand the signatures and the cellular interactions that underlie this good response to SARS-CoV-2, we may be better informed for generating interventions. And so finally, all vaccine candidates and therapeutic targets must be tested within appropriate experimental models. And these need to recapitulate the broad impacts of the SARS virus on diverse tissues. So we're partnering with multiple groups that'll help and helping characterize the natural infection across different platforms and model organisms. And so finally, it's an honor for us to be able to really present this data to you today, but we represent just part of a really large collaborative community within the Broad and across multiple institutions and hospitals in the Boston area. We're also part of this larger kind of worldwide collaboration to fight COVID-19. And so I just want to first take this moment to thank all of the team members who have and really continue to work tirelessly during this unprecedented time to make this work possible. And finally, we give our sincere thanks to the study participants and their families. And thank you for your time and attention.
Wonderful. Thank you to all the speakers, to Chloe and Toni and Carly. What a tour de force effort. I'm sure there are a lot of questions out there. I'll encourage all of you in the audience to ask questions through the Q&A button on your screen. There's a few starting to roll in. Let me ask the first one from Matteo Gentili. He says, coronaviruses do not have a nuclear phase in their replication. Do you recover SARS-CoV-2 RNA in single nucleus data? I think that's a question for Toni.
Yeah, so we do. And it might be surprising that we can detect these viral reads for the exact reasons that you mentioned, but our group and others have shown that single nucleus RNA-seq protocols actually capture RNA from ribosomes that are attached to the nucleus, also RNA from intact ER, as well as cytoplasmic RNA.
Thanks. And the next question from Steve McCarroll, who says, great work, that's high praise, and says it's striking that it was striking in one of the figures that there was not a strong correspondence between the ACE2 expression and presence of viral RNAs. What do you make of that lack of correspondence? And also, are the viral RNAs detectable in the nucleus?
I can answer that. So
great.
Yeah, the lack of the majority of viral RNA positive cells are really surprisingly in the myeloid population. And so this, we assume at this point that this is macrophages at very advanced COVID-19 pneumonia doing their job. Clearing infected cells, engulfing dying or infected epithelial cells within the lung. We do find SARS-CoV-2 viral RNA positive cells within the epithelial compartment and also within the endothelial compartment. The exact overlap of ACE2 positivity isn't very high there. We are not sure at this point if it's because ACE2 is kind of a hard RNA to capture baseline in some of these tissues, and if it's a dropout issue, or if this is some real biology in kind of really advanced COVID pneumonia.
We also think that, you know, the cells that are ACE2 positive and TMPRSS2 positive and infected may just be dying. And so maybe we're not capturing those, especially in these samples with very long PMIs.
And perhaps to add to your point, Toni, right, when you and Carly do the dissection of the tissue, you try to avoid the necrotic part of the tissue. Otherwise, no good data would be generated.
Exactly.
Okay, so third question, is there evidence that the disseminated issues are of a viral nature, or could this be due to self-reactive cells? Is there always viral RNA where there is cell damage?
I mean, I'll answer.
Yeah.
I'll offer one answer and then invite others to add to my answer. So I do not believe there's always viral RNA when there's a tissue damage. There is, in some cases, some very strong innate immune response. There's a hypothesis that proptosis may be involved in some case, right? And so patients are suffering from lymphopenia, the severe patients, and the question is, where are the immune cells? Is it that they're trafficking in the tissue and contributing to tissue damage, or are the cells dying because of the very intense inflammatory milieu? So there is definitely a strong innate immune response component contributing to the tissue damage. But obviously more analysis needs to be done to pinpoint to the actual pathways and self-involvement. This is within the scope of the work that was presented today.
Yeah.
It should also mention that most of the analysis is ahead of us this summer as we spend
you're
the time
really just getting going.
The frameworks, yes.
Absolutely. Yeah, I think a lot of us working on COVID can appreciate just the effort in the logistics and setting up, you know, the pipelines to get this work going, and are looking forward to a busy summer of understanding what's going on. Yeah. Great. Okay, so the next question comes from Troy Whitfield, who says, was the consensus SARS-CoV-2 sequence the same for all subjects, i.e. were they infected by the same strain? A question that speaks to my heart too. How might the answer impact the reference metagenome that was used for mapping and also the interpretation of the results? So are there differences in the viral, genomic genotypes that you're detecting?
So yeah, I'll offer an answer to that.
Great.
One of the kind of the data that we presented today is all aligned to the same viral reference genome, and we're collaborating with people from Party Sabetti's lab to help us do some of this kind of full-length viral genomic sequencing.
Yes.
We actually submitted just some samples to them yesterday to do that. And so the goal, the diversity between different SARS isolates, at least when we've looked kind of at some of those in Massachusetts, aren't radically different, such that we think that we're at least able to pick up with decent confidence many of the transcripts that are cell associated. We anticipate that when we actually can match the specific viral strain from that lung tissue, when we do our alignment, we may actually get an increase in sensitivity into viral transcripts within that. And we also ideally will be able to then reconstruct viral sequences from single cells. And so there's probably definitely some interesting analyses that can happen with that data.
Yeah, I'm involved in that work actually, and we're looking forward to it too. Yeah, so we'll have to do a follow-up in the fall to share those findings. Great. Okay, and the next question from David Gifford. Do you find that 3' amplification of SARS-CoV-2 RNA from subgenomic transcripts that are made of the primary full-length viral RNA? This would potentially imply a bias towards protein levels of the 3' genes in their MHC display.
So we do find 3' bias, but that is most likely due to the protocols. They're 3' anchored single cell RNA sequencing and single nuclease protocols. And so we see bias there. My guess is just because of the proximity to the poly-A and the size of these libraries, and that they're, biased towards that end. We haven't yet done these really important next steps into trying to map out the specific subgenomic and the different species of viral RNA that are in each of these cells. It'll definitely be a challenge from the shorter read single cell RNA sequencing data, but certainly something we'll be doing.
Yeah, we're also going to start to try to incorporate 5' single cell RNA sequencing approaches, also full length sort of bulk RNA approaches too.
I know we have Orit and Alex on the line. Before we carry on with questions, Orit and Alex, is there anything you guys wanted to chime in with? Any comments or perspectives from your end, or should we just carry on?
Good, Orit.
Sorry to put you on the spot, but if you have anything, you're certainly welcome to share it with the group.
So I just want to emphasize again that this is very much a team effort across the Boston community, and we want to thank all our collaborators. There are many of them, and we're very excited to see the results of this work and its implication on this disease.
And I'll just echo what Orit said and also highlight that this is a time where, like other times, but even more so where we'd love input and feedback from the community, because I think we're all trying to work together to address this terrible pandemic. And some of the great questions that have been coming in, are things that I could imagine guiding some of our conversations going forward. So if you're interested in some of the data and some of the analyses, that we alluded to today or that we discussed in detail, we'd love to have further conversations and figure out how we could work together to accelerate our understanding of this disease and develop better cures and preventions.
I would also say that we're intending to post the data as soon as we can, so people can continue to ask questions in the realm that's of interest to them if they have specific questions or specific genes that they want to follow up in these tissues and in the blood. So I think that hopefully would be very useful to the community.
And to add to Orit's point, that it's not just for the autopsies, but for all the projects, including the acute cohort that was described today.
I have a quick question. We hear a lot about animal models and the lack of animal models for studying SARS-CoV-2, but is there any thing, you know, on the horizon, anything that looks interesting that could give you access to this, you know, type of information in more accessible models, or nothing yet that quite compares to getting human tissues.
Chloe, do you want to take this one?
I was going to say, Alex, you should take this one.
I'll let Carly take it. She's done a lot of this work. Good, Carly.
Sure. So it's a really important point. So what we're partnering with a couple of different groups across Boston, and there's a lot of work going on to apply some of these same approaches of, you know, broad single cell genomic assays across natural infection. And what this also gives us a window into is kind of the time vector in a lot of these diseases. You know, in all of this, we're looking at just a snapshot of the most severe,
right,
unfortunately terminal cases of SARS-CoV-2. And some of the animal models that we're kind of helping profile include hamster, include non-human primate. Those are kind of just among the ones that are, at least from my understanding, kind of emerging as some of the favorites. Ferret as well. And so some data from those efforts will also be released in the same kind of an open way as these in the coming weeks and months.
I'll just add a little comment on the back to say that, you know, figuring out what is an appropriate model for different aspects of disease is not something that we have 100% clarity on. It seems like some of the small animal models, like the Syrian golden hamster, recapitulate you know, severe disease, whereas some of the non-human primates look more like, you know, healthy individuals who recover. And I think that what we're going to have to do is piece together a number of these models along with other systems such as organoids, labs on a chip, organs on a chip, and cell lines to try and figure out what interventions, both vaccines and therapeutics, we should advance to additional organisms, and then figure out how to move those into humans as rapidly as possible. So I think really it's going to be piecing together all these different elements to understand why it is that some of the models respond more favorably and some respond a little bit worse, and to understand whether or not actually at a molecular level and a cellular level, the models are behaving poorly for the same reasons that we're seeing severe disease in humans. And that's something we're not 100% sure of yet.
Yeah, so the differences could be meaningful in understanding what's going on in humans, in fact.
Correct.
Cool. Wow, well, we are right on the hour here. There's a few more questions in the Q&A, but hopefully the team here can follow up to answer these great questions offline. And the very last comment from an anonymous attendee was, good luck. It was a great presentation. I'm sure that could have come from anyone out there on this on the line. It was indeed great, and best of luck with the work. We look forward to hearing more about it in the coming months. Thank you all. And thanks for tuning in to everyone out there.
Thanks for having us.
Thank you.
Thank you.